So I'm actually presenting a particular project, but I'm also presenting a concept, which is during outbreaks, it's actually really hard to do research on patients. Biomedical research requires multiple researchers to be able to safely get samples from patients, competing with clinical care, competing with healthcare workers. And so over the last year and a half, we formed the COVID-19 biorepository. Katrin was was a program manager on that for about a year. And so this project is sort of a proof of how you can collect agnostic samples from patients during a pandemic and potentially elicit a particular idea, or test a theory that could lead to future funding. And so what we particularly looked at was actually the question of autoimmunity in SARS-CoV-2 infections. And there's a lot of text, but I'm going to tell you if you look at me, I'll tell you what we're looking at. COVID-19 disease is defined by a couple of things. The impact of the virus itself is defined by hypercoagulability, and it's defined by a maladaptive immune response. And research so far has shown that in patients who are particularly severely ill, you see a huge increase of autoimmune antibodies. And you're also particularly seeing autoimmune antibodies such as lupus anticoagulant that can cause hypercoagulable states, that can cause those blood clots that end up having sort of long-term impacts. And you are seeing also evidence, a study that was presented about a year ago, that there are autoimmune antibodies against components of the human immune system, such as type 2 interferon. And when you saw that, those autoimmune antibodies were actually correlated with severe disease as well. Separately, there's been a question about the fact that people who survive SARS-CoV-2, convalescent individuals, are continuing to have symptoms. 1/3 of people in the outpatient setting, as well as 2/3 of the patients who were admitted at 6 months, still had persistent symptoms, what we are now calling post-acute sequelae of SARS-CoV-2 infection, or PASC. The trouble with PASC has been it's really been hard to control for the definition. And NIH recently funded the RECOVER cohort, which is a national cohort of trying to identify patients who had acute illness and who are now presenting with symptoms, and those who are not, as well as controls. And I'm happy to announce that Boston Medical Center is one of the sites as part of the RECOVER cohort, and I'm the PI for that cohort here at Boston Medical Center. What we did was we actually took those bank samples from our biorepository as well as the MGB biobank on COVID-19. We wanted to answer the question of longitudinally, right? There are many viral infections that cause autoimmune reaction, antibodies that attack your own body versus the virus, because there is some similarity between the targets on the virus compared to similarities to aspects of your body. And it's never been clear what the role of those autoimmune antibodies, which are not high enough potentially, as the same as being, let's say, lupus or scleroderma, which are autoimmune, conditions to begin with. What do they do? Do they do anything? Do they represent some sort of disease? Is that the basis of what we have been calling chronic fatigue syndrome, or ME? And so what we wanted to do was look at patients who are coming from pre-pandemic, a couple of autoimmune diseases, as well as inpatient and outpatient. And what we saw, very briefly, is that when you looked as a group, when you looked at people who are acutely ill or people who have recovered from SARS-CoV-2, compared to pre-pandemics, they were statistically, in some cases, not for all autoimmune antibodies, but in some cases, had higher autoimmune antibodies than those who were individuals from before the pandemic. And in some of those cases, those autoimmune antibodies were actually induced in the middle of their acute illness. So in about 18% of people who are outpatient and about 63% of the people who are admitted for their disease, new autoimmune antibodies were created. So what we're trying to answer is, well, that's okay. The next step that we have to do is actually see from the cohort that we were just funded with to identify whether we can link autoimmune signatures to basically people presenting with long COVID. And then we're going to look at also the inflammatory markers that are related to that. So I will stop there. I'll thank the CTSI for the pilot grant that helped us do this research on the biobank samples.